We are going over the digestive system, chapter 23. Okay. Uh, the overall idea of the digestive system is obviously breaking down the macromolecule into a smaller component. So here's the big picture. You have large material. These are called macromolecules. Digestive system allows you to break it down into a smaller component. These smaller components now, it can be broken down further a little bit later on to produce energy, like in glucose that we discussed, or they could be having other usage. But in order to get absorbed in the small intestine, they need to be small. So when they are passing through the small intestine, they could be picked up by the villi, and in here you had blood vessels, and majority of the substances are getting absorbed through the blood vessels, with the exception of the lipid. What are the macromolecules? Macromolecules refer to as large molecule. So let's go ahead and take a look at four different macromolecules that you wanted to think about. So when we're looking at the digestive system, I wanted you guys to think about the food that you eat. All right, say for example, what you had for breakfast, or what are you gonna have for dinner, right? Say you will have a hamburger. A hamburger consists of hamburger buns, which is mostly carbohydrates. Carbohydrates, again, you did the experiment, so you have some ideas about it. Also, the carbohydrates are known as polysaccharides, means sugar, foods that are rich in starch, potato, banana, rice, noodles. Those are good source of carbohydrates. They need to be broken down into simple sugar. Main source of sugar or the simple sugar would be the glucose, for instance. All right. So along the way, you may need an enzyme in order to facilitate this, to make this happen quicker. All right. And the enzyme for it is called amylase. There are two sources of secreting this enzyme. One would be salivary glands that you learned in anatomy. If you remember, there were three salivary glands: parotid, sublingual, and submandibular. And they can those are collectively known as salivary glands. Salivary glands produces salivary amylase. So the easiest food to break down, or macromolecule to break down, would be carbohydrates. In fact, it won't take you that long in order to break down the bread, for instance. I don't know if you ever done this or not, but if you chew on a bread for a very long period of time without swallowing, you might actually feel the sugar, all right, because eventually the sugar will be released. All right. So those are the carbohydrates. Uh, as you know, when you are breaking down the carbohydrates into simple sugar or glucose, glucose is a source of energy. We discussed the role of insulin and how they can remove excess amount of glucose or sugar into the liver in a form of glycogen. So again, you'll be converting simple sugar back into the form of carbohydrates in the liver, and you could store them. Again, you could break it down and make the simple sugar as you are, needing that particular source of energy. So collectively making and breaking, or anabolic and catabolic reactions, are known as metabolism, metabolic reactions. So again, it's kind of like you have excess money, you deposit it in bank, you need money, you take it out. It's give and take, give and take, give and take. When you're looking at the course of the year, most likely you'll be pretty much same as you started before, at least for the majority of us. Now, the rest of the imaginary hamburger would be the meat. The meat consists of protein. So protein can be broken down, and you already know this because we discussed about the hydrolysis and dehydration reaction on chapter 3, actually chapter 2. And amino acids is when you break down the protein completely, you're going to get amino acids. Chain of amino acids was making up the protein, and we discussed about this type of amino acids. We learned how you could put in this amino acid to make the protein in the process of translation, if you recall. So when you break down the protein, that's what you're going to get. Um, these protein, you may use a different type of enzyme, but in general is called protease. Source of proteases varies. You could have a type of a protease in the stomach that is called a pepsin, all right, which at the presence of hydrochloric acid, pepsinogen, get activated and becomes pepsin, and now pepsin can break down some of these protein uh, peptide bonds. There is also other such as trypsin, chymotrypsin, carboxy peptidase, and a variety of other ones. And the rest you could think of that they are being secreted from the pancreas, with the exception of the pepsin, which is in the stomach. The rest is basically coming from the pancreas. So uh, there is no protease in the mouth. So protein, as you break down uh, or making a hamburger meat more smaller by chewing, breaking it into smaller pieces, that would be a mechanical digestion. But then, you swallow, and the food from the esophagus goes into the stomach, and the stomach is very acidic. So pepsin is going to act, but it's not going to break down all the peptide chains or bonds. So protein moves from the stomach eventually, or what's left of it, to a small intestine, and the pancreas releases its content into the small intestine. And finally, you get a complete breakdown of the protein on the small intestine. The small intestine then absorb these amino acids, and now amino acids going to be in your blood. All right, now from the blood, it can go literally everywhere, including any kinds of the cells that wanted these amino acids to make their own protein. All right, so the third type of macromolecules is generally lipid, and a major part of the lipid in our diet is triglyceride. And the lipid, you need an enzyme to break it down that's called lipase. Lipase breaks down the lipid into a smaller component that would be glycerol and free fatty acid chain. And again, absorption of the lipid is a little bit different. Uh, Lipid is a non-polar substance, so they kind of like coalesce with one another, they stick with one another. First, you need to separate them into a smaller component. But because they are non-polar, and non-polar substances, they like non-polar substances. What happens is you need now a way to, uh, separate them. So therefore, uh, in the liver, uh, bile being released, and the lipid plus the bile, they are now separated. So think about this as being the lipid now plus the bile, they're getting smaller, right? Now lipase can act upon this and break it down into a smaller component, so that way your lymphatic system can take care of that. Okay, so they don't go to the blood. As you know, if they do, then they can clog up the arteries, and that would seriously cause a problem. And some triglycerol do that, unfortunately, as a part of our diet. And they adhere to the arteries and cause cardiovascular problem. Next up, we go over the last macromolecules, which would be the nucleic acid. Nucleic acid uh, is basically the DNA and RNA. So your hamburger contains, I don't know, 30% fat or lipids, but also anything that comes from the live organism. This includes cells that making up the meat, ground beef, for example, the lettuce, the tomatoes, the bread, everything that is coming from plants or other animals and so on. They have DNA and RNA. And DNA and RNA, they stuck with one another, so they're still a large molecule. And in order to break them down, you need an enzyme. And the enzyme, again, is called nuclease. And it breaks it down into nucleotides. Nucleotides is basically a smaller version of the DNA and RNA. And you probably know they have a sugar base, they have a phosphate, and they have one of those ATGCs or AUGCs. Regardless, you make those. Now, I wanted to kind of like highlight all these enzymes. These enzymes are being released by the pancreas. And if it says pancreatic amylase, half of it or some of it is by the salivary amylase, but the rest is from the pancreatic amylase. Therefore, one of the function of the pancreas is to bring and release all these enzymes for our digestion. If your pancreas is weak, if it's not producing the enzyme that it needs, again, you might say, wait a minute, how is the enzyme is being made in the cell? Enzymes are protein. So again, you got to go back and review what we discussed earlier and see the overall picture. Right? Protein made out of amino acids. That's translation. So pancreatic cells that are known as acini, right? And you learned it in the anatomy, that's where they are being made, all right? So we discussed the cell already. Inside of the cell, you have DNA, then you can manufacture messenger RNA, then you can make protein, right? In this case, the protein that is being made are a variety of different type of enzymes. And once you get the enzymes, you get to break down the large molecules. And majority of this happens in the small intestine, but we already know that that happens in the stomach using the pepsin. And this, at least some portion of it happens in the mouth. Okay, but other than that, uh, everything else, the location would be in the small intestine, and the enzyme that is being released in the small intestine to be able to break down the macromolecules happen to be from the pancreas. So that was just kind of like an overall picture. Now, a lot of these, you guys, if you haven't done so, like, carbohydrates, that was an experiment related to that. How do we get the sugar into the small intestine and so on. So there are some experiments related to that we discuss. Later on, we're going to talk about the diabetes and so on, probably tomorrow, and you get ideas about that as well. All right, now that you know the overall picture, again, we wanted to go ahead and take a look at the compartment in which the food travels. In anatomy, you learned that about those compartments. Today, we're going to be focusing more like what are those compartments do in terms of physiologically and helping us in terms of their function. All right, so let's go ahead and take a look at stages of the food processing. One of the things that you need to know is the 6 stages of the food processing. In other words, as you place the food in the mouth, all right, that's where the ingestion is occurring. All right, so let's go ahead and take a look at it. This is ingestion, placing the food in the mouth. All right, now awful lot of things happens in the mouth. One is the mechanical digestion. So this is another stages of the food processing. Mechanical digestion is basically grinding, chewing, turning, you know, making things smaller mechanically, movement. All right, so your teeth coming down on the food, all right, crushing them, making it smaller, and those are mechanical digestion. All right, very little absorption happens in the mouth, okay, but chemical digestion also begins in the mouth. But what is the chemical digestion? Again, using enzymes, there's a chemistry involved, there's a chemical reactions involved. All right, enzymes are in the chemical reaction help to, you know, get to the product from the reactant or the substrate to the product much quicker. Let me mention about the role of enzymes. All right, so this is where it happens in the mouth. Chemical digestion begins in the mouth. Mechanical digestion begins in the mouth. Ingestion begins in the mouth. So, but the chemical digestion is not going to be that sufficient. For example, uh, the salivary amylase that produced and released by the salivary glands only can break down the carbohydrates. Okay, there is a little bit of lipase in here. It's called a lingual lipase, but again, very very little, not a whole lot for our purpose. Right. So uh, amylase is the main one, and if you wanted to take a look at the salivary glands and such, you could go ahead and take a look at this picture. So it could tells you where they are: submandibular, sublingual, and parotid gland. And again, if you look at this duct, they're all going into the oral cavity where the mouth is. All right. So they release their content, and their content is going to do few things for us in the mouth. One is called salivary amylase, break down the carbohydrates. Two, salivary amylase and the saliva itself, it can lubricate the food. When you lubricate the food, it's easier to ingest and swallow. Okay, the food is lubricated, right? So lots of things happening, but in the nutshell, you know, your saliva can cleanses the mouth. It can moisten and dissolve the food chemicals. Remember the taste, they require molecules to get dissolved, so it aids that. Aids in bolus formation. Bolus means just like, you know, a smaller version of the food so that you could have it easier to break things down. Contain enzymes that break down the starch or the carbohydrates, all right? And that is what it does. Now, interestingly enough, when we're going back over here, and we mentioned amylase is important. That's the role of the enzyme. Interestingly enough, if you go back to the lecture that I was giving you about enzymes, we mentioned there were different uh, factors affecting the enzyme function. One was temperature, one was pH, and one was salt concentration. All right. So right there, you had a good idea. Just we are inside of the mouth at this point in time. You're biting on your hamburger. We haven't gone to the other parts yet. Right. But what do we have a tendency to do during eating? Let's say you're going and buying in and out drive-through. All right. You usually get a fries with it, and you usually get a Coke or some kind of a soda with it. Coke in the hot summer days is really, really hot. All right. So the Coke is cold with ice in it. So right there, you're influencing the temperature. Your fries also contains a lot of salt. Some of us put extra salt on the fries because usually food that are salty tastes better. All right. So a lot of sodas are actually acidic. All right. So you have all the components that's supposed to influence and mess up our enzymes in the mouth. So it's not a good idea to have salt. It's not a good idea to have soda with your meal. All right. And beside these, uh, whenever you're eating soda, you're eating your meal with the soda, you're consuming soda with it. What else it can do is it prevents your saliva to be released because it doesn't need to lubricate the food anymore because you're getting the lubrication from the soda that you're drinking. Here's the a kicker. Over long period of time, all right, uh, not using your salivary glands, they shrink, they get smaller with aging. All right. So as a result of that, now you are more dependent on these sodas for your consuming of your meal because now every time you wanted to have it because you don't get the lubrication and the moistening of the food and the dissolving capability of it. So you become more and more and more dependent. It gets to a point that your mouth always feels dry. All right. So again, it's not a good idea to have, uh, you know, drink with your meal, maybe 30 minutes before the meal, maybe 30 minutes after the meal. And certain culture usually have a hot tea, for example. Hotter substances usually helps with the dissolving of the material, and the cold materials and so on, it makes it more difficult. You could do a simple experiment by, putting some kind of a oil that you have in the cold environment, see what happens. They solidify, all right? If you have maybe your dirty pot with oil in it that you use to cook something overnight, the next morning you notice that it solidified because at nighttime it was cold. So again, you notice all that, okay? Uh, the temperature that you needed mainly it's supposed to be a gut temperature. It's supposed to be about 37 degrees C. That would be the ideal temperature. All right. When you're having a cold and so on, eventually it's going to get there because it goes within the body and the body core temperature is 37 degrees C. So it gets it warm, but you're just adding a little bit more stress and more pressure, and the enzyme is not going to be as effective and digest the material as fast. All right. That doesn't mean that it doesn't happen. All right. So next thing we wanted to talk about is propulsion. All right. Uh, Propulsion is swallowing and idea of peristalsis. So as you know, when we swallowing, a couple of things happen. By the way, no digestion happens in the esophagus. Esophagus is the connection of the mouth to the stomach. So over here, there is not really a particular way of, the the layers and so on. They're not set or they're not structured for, absorbing anything. Okay. What happens, it's just a connection. It's a tube that connecting the oral cavity or the substances that you eat towards the stomach. All right. So what happens in the esophagus? First, we need to uh, go back and take a look at peristalsis. So if you have a bottle of water halfway filled with water and you squeeze towards the bottom, the water goes up. You squeeze the top, the bottom will go down. If you have, for example, here's your water bottle that is flexible, all right? So you could squeeze in here, the water will go the other way. You squeeze in here now, the other water is going away. So these ideas of going back and forth, these are called the segmentation. And the idea of peristalsis is contraction and relaxation of the smooth muscle allows the bolus of the food to travel. So right here is a contraction, right here is the relaxation. So now they start contracting right over here again. So you could see what happens. The food is ending up going downwards. All right. Peristalsis can happen variety of places. That is from the esophagus to the stomach, from the stomach to the small intestine, from the small intestine to large intestine, and so on. Peristalsis actually can actually occur in our reproductive system too, but you're not processing the food. You might be processing the sperms or eggs, and we'll talk about that a little bit later on. All right, but that's what peristalsis is: movement of the food, processing them by way of contraction and relaxation of the smooth muscle. Let's go ahead and take a look at the four layer in which you needed to know. And your anatomy instructor probably went there already. So doesn't matter, esophagus, the stomach, intestine, a small intestine, large intestine, or colon, you have a lumen. Lumen is where the food is passing through, all right? Then you have four layers. You need to know these layers. First is the mucus, then is mucosa, then is the submucosa, sub means below, below the mucosa, and then there is a musculatura externa, which is again a smooth muscle, and then the last one was the serosa adventitia. All right, these are smooth muscles. Consist of majority of it longitudinal and circular. If we're talking about the stomach, you have an oblique muscle as well. But these muscles are really, really strong, and these muscles are participating mostly in the action of peristalsis that we discussed. So even if it's in esophagus, these muscularis externa, again, via the sympathetic and parasympathetic fibers, if we are in the parasympathetic, they start, contracting and so on and processing the food, and therefore, digestion is happening. All right. And what happened is, they are participating in the peristalsis action. All right. Moving right along. We are now inside of the stomach. All right. In anatomy, you learn that this one is probably, your gastroesophageal sphincter or cardiac sphincter. It opens up. Now the food is inside of the stomach, and food can retain in the stomach, remain in the stomach for up to about many hours, 6 hours or so. Majority of what's happening in the stomach is due to the cells in the stomach that produce hydrochloric acid. All right, those cells are known as parietal cells. So let's dive inside of the stomach. All right, we'll look at the idea of the different type of cells that they're there, and then we will take a look at the physiology of what is happening in a nutshell and making it simple and such. So let's go ahead and take a look at the stomach itself. All right. Before we go to the stomach, I wanted to point out another important thing that is happening, and that is the role of epiglottis. This is the epiglottis right here. Normally, the epiglottis is up, so the food can go in or out, no problem. I'm sorry, or you could breathe. Air can go in and out of the trachea, no problem. All right, so this is the airway. This is the trachea, I think is labeled right here. All right, so behind the trachea, as you learned in anatomy, lies the esophagus. This is behind or posterior to the trachea. So we don't want the food end up in the trachea and it can block the airway. Now you won't be able to breathe. If, for example, the food is stuck right over here, now the airway is blocked. This is when a person is choking, by the way. All right. So we do not want this to happen. So, the epiglottis comes down and blocks the opening to the trachea, so the food or the bullets of the food have no choice but to end up in the esophagus, which again, you'll see that. And furthermore, you could see the action of peristalsis right here, all right, and therefore the food is ending up in the stomach on the very last part. All right. So these gastroesophageal sphincter, normally they are closed. They will open up and therefore the food can end up in there. Okay. So now we are going inside of the stomach, right? So if we go into the stomach, we'll see that again, a little bit of background information. You need to kind of like take a look at your anatomy perhaps, but I don't think I have this slide here. But that's okay. Let's go ahead and take a look at the inside of the stomach. Here is inside of the stomach. All right. If we look closely, you see the stomach. There are cells in here. All right. But these cells, there is like a smaller indentation in here, and these are called the gastric pits. All right. So the gastric pits, if we go ahead and take a look at the gastric pits and kind of like review them a little bit larger, again, you see kind of like cells that are going really, really deep. All right. And here is the lumen of the stomach. All right. Again, the cells are going really, really deep. All right. And I'm going to make it simple, but basically, this is the lumen. Same thing as here. So these are the gastric pits. This whole thing is called the gastric pits. And these are made out of cells. So there are cells in here. Every now and then, you see a larger cells. The larger cells are known as parietal cells. And they have, very nice nucleus. If you remember, nucleus was round, larger cells, and so on. All right. So at the beginning, you see cells that are near the opening of the lumen, and these are the known as mucus neck cells. All right. The blue cells happen to be parietal cells. For us, this is pretty important. Okay, and then towards the bottom, you see some cells too. And I'm not going to draw all these cells, but you could just imagine that there are a lot of these cells nearby, and every now and then, again, you're getting, you know, these are supposed to be the cells. You can take a look at pictures on Google if you like, and these are known as chief cells. All right. So, and then there are other cells, mostly are these cells that we discussed, but the mucus uh, neck cells and the mucus cells is basically they are important because they, as the name of it implies, they pretty much produce soluble mucus. All right, and it is really not quite well understood, but this function is perhaps maybe to protect the lining of the stomach itself. All right. Parietal cells produce hydrochloric acid, HCl. All right. So it goes into the lumen. So HCl, hydrochloric acid. And this is important because it can cause a precursor of the active enzyme, which is known as pepsinogen. And these chief cells, they produce pepsinogen. So I don't know if we have room. So pepsinogen is going to be released as well, but pepsinogen only become pepsin in the presence of hydrochloric acid. So chief cells produce pepsinogen, uh, parietal cells produce hydrochloric acid plus hydrochloric acid. Now this becomes pepsin. Pepsin is a type of a protease, as I mentioned to you earlier, and obviously they are important in breaking down the, you know, the meat or protein. There is a little bit of chief cells also secrete lipase, but again, it's very, very little, and we're not going to, elaborate too much about that at this point in time because the majority of it happened to be by the uh, pancreas that I just told you earlier. Okay, so there are a lot of chemical messengers that are also produced by endocrine cells, things in the nature of gastrin, for example, which is a hormone. They play an important role in regulating the stomach secretion and motility, all right? And serotonin, histamine, somatostatin. So there are other type of, you know, endocrine activities there, and each one has its own story, which we probably not going to get into at this point in time. But the point is that the stomach is pretty acidic. Now, before we go on and take a look at how this acidic material uh, mechanism of production of these acidic material or HCl, um, I want to give you an idea. It's very important, by the way. Here's the esophagus, here's the stomach. See? The stomach has to have hydrochloric acid because, again, this is necessity for breaking down the protein. Majority of the protein, they require to have pepsin, all right? So for whatever reason, if you do not have the acidity that requires in the stomach, first, the protein digestion might take a longer time, all right? Might take a longer time. You might have abdominal pain or problem in order to breaking down the protein. Every time you eat something meat or protein, you get a stomach ache. All right. But in addition to that, the pH of the stomach is about 2 to 3. Right? That's pretty acidic. All right. And when you are bringing your food, all right, you might be bringing other things with it, such as other bacteria, such as viruses, such as microorganisms. But majority of these microorganisms, they cannot withstand this acidic condition. So they can get eliminated, which is again another interesting fact that you're kind of like, if hypothetically, if there's an important meeting or there's a courthouse or somebody, they usually search or they have a metal detectors, you have to go through if you have any weapon or gun or whatever, uh, they will stop you before that person is going to cause trouble. All right. So before all these microorganisms causing trouble, they're going to get eliminated right here. This is not to say that not all the bacteria are like that. Like, as you know, Helicobacter pylori is one of those that could cause ulcer of the stomach and cause problem for us. All right. And they can hide, and they do have their own mechanism of withstanding this acidic condition, which we're not going to get into at this point in time. But in a way, it serves us very well. But sometimes people that have a stomach ache for no apparent reason. And sometimes they don't even know why they got a stomach ache, but they think that their stomach is too acidic. Maybe the lining of the stomach is irritated due to consumption of the acidic food and so on. So they cannot tolerate that. And what they take antacid. What's antacid? Opposite of the acid. Basically, is a base. Acid and base. Remember, the base produce hydroxide ion, and the acid was producing, or a proton donor, hydrogen ion. So together you're making a water if you have the similar concentration. So if you have hydrochloric acid in the presence of the solution, it becomes H plus Cl minus. But the more of this you have, the higher the acidic would have. So when you're taking antacid, basically you're introducing some kind of a hydroxyl ion, NaOH or some other kinds of whatever, and now you're making a water because this too gives you a H2O, and as a result of that, making it neutral. All right. As far as the making things neutral, this could be problematic. Why? Because remember, the microbes now in the neutral environment, they could they could survive. All right. Now what? Now they could go into other areas such as the small intestine, such as large intestine. Again, uh, things like bacteria can, can double up in 20 minutes. So very soon they're going to have more and more and more of them available. As you know, a lot of these unwanted microorganisms, they might cause problem for us, such as diarrhea, such as, perhaps even colon cancer. And there's a lot of other problems that are linked to this microorganism, and it's not a good idea to have them. All right. So having an acidic environment in the stomach is actually a good thing. For whatever reason you have this problem, abdominal pain, stomach ache, whatever, you have to find out the underlying reason to try to prevent that from happening, but not taking antacid. That's not a good idea. Antacid also cause other problems such as constipation and dehydration and other problems as well. So there's a lot of side effects. We are going into the hydrochloric acid. Uh, Earlier, I showed you that the parietal cells are producing the hydrochloric acid. Let's see how. So let's just draw a parietal cell. Here is a parietal cell, for instance. Parietal cells were kind of like uh, they were surrounded by chief cells, so this would be a chief cells on top and at the bottom. All right. So this part right over here would be the lumen. So think of it where the lumen is, open space. And inside of your stomach, there are different lining, including the blood vessel. Here is the blood vessel in one of the stomach wall. There are a lot of blood vessels, as you remember in anatomy. Okay? So now, what happened in the parietal cell is carbon dioxide combines with water, all right? And there is an enzyme called carbonic anhydrase, all right? So there is an enzyme in here. This enzyme is called carbonic anhydrase. You saw this enzyme in the respiratory system as well. So with the presence of uh, carbonic anhydrase, what you get is carbonic acid, which is H2CO3. And H2CO3, what happens is they can dissociate to bicarbonate. Bicarbonate is HCO3 minus plus hydrogen ion. This is bicarbonate. Bicarbonate, if you remember, it was a buffer, and we discussed this buffer system a long time ago. All right, in the chapter 3, I believe. All right, so now what? Now, what happened is this, there are channels in here. There are channels in here, and this is what's known as chloride shift. So one HCO3 goes out, all right, and one chloride ion comes in. So Cl is a negative charge, HCO3 is a negative charge. So in terms of electronegativity, is going to be the same, right? Electron neutrality remains. It's not going to change in this case, all right? However, there are channels for, these are leak channels for chloride ion. So as the chloride ion increases, it's going to go to the area that is decreasing. That would be in the lumen. This is facilitated diffusion. You're going from high to low, all right? So now chloride is in the lumen. Now we got to figure out how it can combine with hydrogen ion. So we need a hydrogen ion. So pay attention, this is a little bit tricky, all right? So let me just use a different color for that. All right, here's a hydrogen ion. The hydrogen ion, right, there is a hydrogen potassium ATPase or hydrogen potassium pump. Earlier you learned about sodium potassium pump. Right? This is hydrogen potassium pump. So what does it do? Well, you're bringing one hydrogen out, hydrogen is plus, and you're bringing in one potassium in. Potassium is also plus. So you need to, this is a pumping now because you're pumping potassium inside of the cell. If you remember, you had higher potassium, So to bringing potassium in, you need a little bit of energy. That's where the ATP comes in from the mitochondria that we discussed and so on. Now, there are leak channels for potassium. So potassium also can leak out again, all right, as they are accumulating, and then bringing it back to inside of the cell as well. Bottom line. The bottom line is the hydrogen was pumped out and potassium keeps coming in, and then it goes back to another channel through the lead channel and so on. But now these two can combine and formulate the HCl that we discussed. Now, this HCl, it can activate the pepsinogen to the pepsin, and therefore you can go ahead and do the digestion or breaking things down. So these are the things that happens in the stomach. All right. Of course, you probably heard about acid reflux when the material keeps going up and so on, and you know, perhaps maybe your lining is irritated, and obviously those things are problem. All right. When the stomach is way too acidic, it tells you something. People sometimes, when they drink too much alcohol or excessive alcohol, they throw up. Why? Because their stomach is getting so acidic that the body is giving you a warning. All right, and that's why it's happening. All right. I'm gonna. All right. So we discussed what happens in the stomach. We are now going uh, away from the stomach, and the stomach, as you know, is linking to the small intestine. So the next thing I like to do is travel inside of the small intestine, and again, learn a little bit about what is happening there, and we can go from there. Interestingly enough, so we went over this already. A small intestine consists of three regions. Uh, the first region was the duodenum, middle one, jejunum, and the last one is ileum. And there are a lot of other things that link to the small intestine. So just to give you an idea, it's a majority of digestion and absorption happens in the small intestine. We discussed about the principle of complementarity and how the uh, villi and microvilli are important in this process. But interestingly enough, we will see. Let me see if I have a picture of that. Uh, I guess I have to draw one. Okay, there it is. All right. So notice like the content of the pancreas, all right, is going into the duodenum. This is the duodenum, the very first part of the small intestine. And if we can maybe draw a liver right over here, all right, this is the liver, all right. So these are the bile ducts, all right. And you can see or hepatic ducts going into the bile ducts, all right. And here's the gallbladder, all right. And look, this is the cystic duct. So all of these content of the gallbladder, the liver, as well as the pancreas, they're all going to go into here, the small intestine. What is their content? Well, so far we learned the pancreas release a lot of different enzymes. We learned that, okay? And earlier I told you that bile was important in separation of the fat. So there is a reason why this is called a bile duct, for example. okay, because that's where the bile is being released. So gallbladder stores bile. So notice that a liver comes in here, but then, you could store some of these bile into the gallbladder, but you could also take the bile and empty it into the small intestine, pancreas, enzymes. Remember, this is the lumen from the small intestine, but actually, where is it coming from? This food going in here is coming from the stomach. And what did we say about the stomach? The stomach was very much acidic. We just mentioned hydrochloric acid. So hydrochloric acid now, plus whatever remaining, the bile or the bullous or the chyme, uh, the remaining of the food, they are coming. Not everything broken down. All right. In other words, yes, some of the protein broke down, but none of the lipids, none of the nucleic acid, majority of those are intact. Still, some carbohydrates might be there too. And a lot of protein might be still there too. Proteins are huge chains of amino acids. So there could be a lot left. So we wanted to break all those things down. That's why there is a pancreatic amylase to break down the remaining of the carbohydrates, if any. That's why there are different type of a protease. Again, I mentioned a couple of them, and they can be inactivated first and then get activated, like trypsin, which inactive form of it would be trypsinogen, or chymotrypsin, in activated form would be the chymotrypsinogen, or carboxypeptidase, inactive form would be the procarboxypeptidase. All of those, they're different enzymes. Majority of it, we call it is a protease, all right? But the point is that they need to get activated. So in the small intestine, there are some epithelial cells that have properties to activate things, and then as a result of their product activating other things, you get a lot of these enzyme activated. But the point is that the enzymes needs to be there, but also bicarbonates. Bicarbonates is HCO3 minus. Although we needed hydrochloric acid to activate pepsinogen in the stomach, but now these enzymes, they don't need such a high acidic condition. In fact, they're going to affect the role of the activity of the enzyme. So what we need to do at this point in time is to neutralize those acidic hydrochloric acid or acidic condition. So HCO3 offers that. So not only producing and releasing enzymes, but also what you're doing, you are neutralizing the content of the stomach, so that way they could actively start breaking things down for you. Now, the liver that produces the bile, so liver produce the bile, gallbladder stores it. Uh, those are accessory glands. Pancreas, accessory glands. Liver, accessory glands. Gallbladder, accessory glands. Okay? There are part of the accessory glands. That means what? That means they help out. All right? In fact, right here it says accessory. Right? So they're there for a reason. All right? So let's go ahead and take a look at the liver job other than the bile production. So one of them, the major role is production of the bile. But they also do other things. A storage of fat-soluble vitamin, detoxification, processing blood-borne nutrients. So they could do a lot of different things. We discussed about the nutrients already, the glycogen and the role of insulin and glucagon. The fat-soluble vitamin are vitamin A, vitamin D, vitamin E, vitamin K. These are fat-soluble vitamins. The rest of the type of vitamins are known as water-soluble vitamins. Now, what's the difference? Water-soluble vitamins, there's no pose or threat in our cells. In other words, when you're taking so much of it, your urinary system can take care of it. For example, water-soluble vitamin would be vitamin C, vitamin B, B12. There are all kinds of other vitamins around, and a lot of those are known as different supplements. And you can purchase those different areas and so on. But or you could have it in the fruits and vegetables and your food perhaps too. Point is, water-soluble vitamins, you can excrete them via your urinary system. There is not much of a problem. Uh, if you take in a daily vitamin, your urine becomes really, really yellow. Why is that? It's because the excess came out. You don't need them. You don't need them to process or absorb them. It comes by waste in part of the urinary system. However, the vitamin A, vitamin D, E, and K, they're pretty important. I mean, vitamin A, that was important uh, precursor to retinol of the eye. The carrot, for example, has a good amount of vitamin A. Vitamin D is good for bone synthesis, as you know, and calcium absorption. Vitamin E for tissue repair. Vitamin K for blood clotting. So they're all important, obviously, but when you take in too much, it can stores within our cells and cause toxicity. So, storing them perhaps would help you in that matter. And detoxification is another important function of hepatocyte. Hepato means liver, cells, cyte means cells. And the detoxification is important because sometimes there are substances in our meal, material that we take in, and they're very toxic for our body. The liver pay the price of detoxification of those substances, and sometimes it's not good for the liver to keep doing that. You know, surprisingly, a lot of medications they can destroy your liver. They don't tell you that so that you could buy them, or perhaps they put a fine prints on them. Uh, Tylenol, if you're taking excessive dosage, it could cause liver damages. And if you have a Tylenol handy, or you can maybe reach out in your medicine cabinet, take it out and read it carefully. Should be a warning sign there, somewhere around there, that higher dosage could cause liver failure. But the liver is made out of hepatocytes, all right? And again, you're trying to doing the detoxification using the liver cells, but your liver cells in the process might get problematic over the long run, just like anything else that you use up so much, so long, they wear out and they won't do their function after a while. And if you have your liver not functioning, you could have all kinds of problems, including not production of the bile, including not being able to detox, including not being able to, do the necessary function, and it could cause major problems. Psoriasis, for example, scar tissues in the liver, all of those people that drink a lot of alcohol, they destroy their liver, all right, completely sometimes, all right? It gets so bad that, you know, the liver is not functioning anymore. All right. So next up, we can go ahead and take a look. Here are the uh, anatomy of the liver. These are the kind of like lobules of the liver, and it just shows where the bile is being released. There are some Kupffer cells. These are kind of like macrophages that again they can destroy, they can engulf, and we discussed about the endocytosis already. All right, things that are unwanted. Moving right along. Uh, if we go ahead and take a look at this particular slide, what do we see? We see the pancreas producing its content. And again, a lot of these are inactive that I told you, and you're making it active, all right? And as a result of that, you could break things down now. So pancreas is important, liver is important. They help out. They're known as uh, accessory glands. In addition to these, if we go ahead and take a look at the small intestine, you'll see there is a lot of foldings. These foldings were called villi that you identify. And again, each of the cells consists of subfolding known as a microvilli. And these are increasing enormous amount of surface area. All right, this surface area, obviously, it helps to absorb substances, especially when they are smaller, because large substances now broken down by help of enzymes into smaller things and so on. So now they could go right in and again end up in the bloodstream, or if it's fatty stuff, it could go into the lacteal, which is ending up in the lymphatic system or whatnot, but the majority of it, it goes into the bloodstream and going into the, reaching out your cells. This could be amino acids, this could be sugar, this could be other things as well. All right, vitamins and such. So microvilli and villi, they gives you a means of high surface area. All right. And there is a lot of chemical digestion here. Chemistry is involved, enzymes are involved, because all of those enzymes released by the pancreas we looked at earlier is being dumped right here in the small intestine. So earlier I showed you a figure, and it's, where is it? Right over here. So there are a lot of enzymes are doing their work. All right, so breaking the molecules into even further a smaller component, so you could do a absorption and the layers. In those layers, there's a lot of blood vessels there that they can get absorbed, and some of those blood vessels you're already familiar with. Look at some of these blood vessels. All right, mesenteric arteries and all kinds of capillaries around and such. Right, so there's plenty of blood vessels. So once it gets absorbed, it can get into the blood vessels, and from there goes into the circulation. Huh? All right. So uh, it's important to know how you can monitor things, how we regulate production and releasing of the bile, how do we regulate releasing of the enzymes, how do we make more of those enzymes and such. So this little bit of physiology involved that I like to get into next. So in order to understand the physiology and the mechanism, first I need to go over a few ideas with you, and then we can see this in action. See, there are various different type of glands. All right, doesn't matter what kind of a glands we're talking about. Different glands. We discuss salivary glands. We discuss pancreas. We can discuss pituitary gland. We can discuss, pineal gland. There are various different glands in our body, as you learned in anatomy. A lot of these glands, thyroid gland, parathyroid gland, right? So they produce hormones. Sometimes many different kinds of hormones, sometimes one. All right. Now, these hormones, they, again, if you remember, the function of the blood was transport of substances. These hormones, now they can be transported via the blood system. All right. And then once you get to a target cell or target tissues, what happens is that these hormones is going to go in and affect this target cell, whatever this target cell is out to do. And again, there are various different type of cells, and they do different things. Now, this target cell, for example, could be another gland that it could produce another type of hormone, or it could be producing certain type of protein, for example, or it could have variety of different ways that this target cell might get affected. But two kinds of hormones. One is the steroid type, and there is another type. It's called the non-steroid hormone. Non-steroid hormone, such as you know many different types, like CCK, like secretin, like growth hormone, like you know uh, thyroid hormones and such. They are uh, need a receptor. So they need to bind to the receptor. That's a hormone now, right? This is our hormone, all right? They need to bind to the receptor, and then it's going to influence the target cell somehow. This could be, again, maybe a G-link protein, or it could open up a certain channel, for example. It could be linked to the ligand-gated channel. It really depends on the type of a cell, all right? However, if you have a steroid hormone, let's show the steroid hormone by red color, and let's say another gland is producing a steroid hormone. The steroid hormones, sometimes they have a carrier protein, all right, they can carry them, and then again, after they come out, they can just go in right through the cell and influence the cell a little bit differently. We're not going to deal with the steroid hormone at this point in time. That would be the topic of reproductive system, but things like androgen, testosterone, progesterone, corticosteroid, estrone, estradiol, all of those, they are a type of a steroid hormone. So the steroid hormone, they are non-polar. They could just go right in and cause the cell to do certain things. So now that I give you this introduction, I wanted to go over the mechanism of showing how the bile and the pancreas are being controlled. Okay? Let's go ahead and take a look at this regulation. All right. So acidic material from the stomach, known as acidic and fatty chyme, coming into the duodenum. But there are some cells on the small intestine that they have capability of production. We call these introendocrine cells. Introendocrine cells belongs to the duodenum. And two of them that you need to know is cholecystokinin and secretin. For cholecystokinin, right now, we are using the CCK. CCK, abbreviation for cholecystokinin, all right? So these two hormones also play important role in regulation of the pancreas. Let's see what they do. All right, so the CCK, after release from the small intestine cells, known as introendocrine cells, all right? So a small intestine, or particularly uh, duodenal, uh, it can do different things. One is to cause the gallbladder to contract, and another one is to cause the hepatopancreatic sphincter to relax. Let's see what they are. And best way to do this is, again, look at the next slide. So notice that from the stomach, this fatty material and acidic material entering the first part, which is the duodenum. Here are the intro endocrine cells. And as you can see, there are two colors. One of them is red, the other one is blue. The red color signifies the CCK. There are blood vessels in there, and again, hormone travels via the blood. I just mentioned to you. So it goes right into the blood and reaching the gallbladder, causing the gallbladder to contract. Contraction of the gallbladder. All right. So going back to our discussion, uh, we are looking at how we can regulate the bile. Where is the bile coming from? From the liver, right here. All right. Is it useful to keep making the bile if you don't need it? No. Is it useful to wanted the bile but not having it? No. So we need to regulate it. So you need it, you get more. You don't need it, you save it. All right. Now, in order to separate the fat, we need bile. Okay. At the very end of these slides, if you go towards the end, you see is a fat globule right here. All right. But because they're all fats or non-polar, they stick to one another. Right. For example, not only you had hamburger, but maybe you had salad dressing, which had olive oil. The olive oil is going to combine with the, you know, your juicy burger, at least the fat parts. Maybe afterwards you had ice cream. Maybe you had some mayonnaise inside your burger. Maybe you had some butter. Maybe you had, all of those are fat, sources of fat, right? So they all stick to one another because they're non-polar. Non-polar attract non-polar group. So you're going to get a lot of fat globule. That means big globule of fat stuck to one another. Here's the bile or bile salt. Interesting thing about the bile salt, it has a polar group and it has a non-polar group. So when you have a lot of bile, what happens? The polar group, the non-polar group attached to the fat part, which is, you can see it in this part is the green part, is attaching to the fat part. And the non-polar group, they don't like the fat, so they're separating from one another. What it does, it produces a small, they're known as a micelle or droplets of fat. In other words, this is called emulsification. The process of emulsification is making a large globule of fat into a smaller droplets. All right, that's what it is. If your pot is oily, all right, what happens if you just try to clean the oily part? Here's your pot. Let's say that there are layer of the oil on it. If you wash it with water, notice that still your pot is oily after that. Okay, so what do we use? We use detergent. All right, when you're using the detergent, detergent is similar properties than bile in the sense that it has a polar end and a non-polar end. So let's presume that I'm adding the detergent. All right, this part, the tail part of the detergent, has a non-polar capability, so it's attached to the non-polar. And the head part right over here, it has the polar capability, so it's kind of like away from that. But when you apply the water, water is polar, right? So again, it pulls the polar region, and guess what? They take out the oil with it, and therefore you could wash your things. And if you look close up, you see tiny little droplets, bubbles forming. when you use a lot of suds, when you use a lot of detergent and so on, you see tiny little bubbles. That's what they are. Okay? So similar to this is the same concept. This is called emulsification. All right? So when we are going and having a lot of fatty tissues, all right, and these fatty tissues from our meal, from ice cream, from whatever, ending up in our small intestine, they stuck with one another. We don't want that. So these cells capable of releasing hormone CCK, which is in this case, these are the CCK, they cause to be released and go into the bloodstream and then finally ending up in the gallbladder. If you remember, the gallbladder consists of the bile. So when you contract it, the bile is going to be released. So now you're going to get more of them because this bile ending up in the small intestine, that's where you want it. That's where all these fatty stuff is entering. Now you could deal with it. You could do emulsification. And again, your pancreas is producing the enzymes, lipase, to break them apart. All right, so that's the job of the CCK, contraction of the gallbladder. But if you look carefully, I'm going to erase all that. All right, if you look carefully, this part is the opening right here. All right, so we need to, there is a sphincter there that is normally closed. We need to open that. Another thing that the CCK does, it makes these sphincter to relax or open up. So when they're open, guess what? Now they could be released in here. So two things that you need to know that what the CCK does. All right. Where does it come from? It's coming from the intestine cells of Dwalin. What does it do? Contraction of the gallbladder as well as relaxing the hepatopancreatic sphincter. So now the bile happened to be in here, which what we want because it can mix with the fatty stuff and does the emulsification. All right, what about the blue one? The blue one is the secretin. All right, so these are the blue one. The secretin is another hormone. It goes into the bloodstream, reaching the liver. And it kind of like tells you what it does in here. All right, let's take a look. All right, so what it does, it can stimulate the hepatocytes to produce more bile rapidly and being able to transport them. All right, release them. All right, that's the role of the secretin. So both of which is important, obviously, but we are trying to see how we could get the help from them. Now, what about the people that have their gallbladder removed? All right, there's gallstone sometimes forms. All right, due to a variety of factors, and there are different theories how gallstone is forming, but I'm not going to get into that right now. But sometimes you need to remove the gallbladder. It's still, the liver is capable of production of the bile, and it's still the bile can end up in the small intestine, and it's still they could do the emulsification. But you have to be very careful of what you eat, because if you eat a lot of fat stuff, you're going to have a hard time breaking it. You're going to get abdominal pain. You know, you want to be in pain and being uncomfortable. It takes a long time, and as might as well have a restricted diet, low amount of fat as much as possible. All right, that's why people with the gallbladder removed, they need to have a restricted diet. All right, so having said that, let's go ahead and take a look at another one, which is how do we control the pancreatic release? Is not advantageable for the pancreas to keep releasing the enzyme, because then they have to keep making the protein. The cell is going to get exhausted. They need a lot of resources to manufacture things. Right? Same thing with the bicarbonate. All right. when you are making so many bicarbonate and so on, that exhausts the cell. Any cell does a lot of work, they can get exhausted at times. So we got to be efficient. In other words, why would you want it to release bicarbonate if you don't have much food in here to begin with? Why would you want to waste all those enzymes if there's nothing in the small intestine? All right. So again, that's where the hormonal control comes in. As soon as the food enters the small intestine, the cells, the endocrine cells of the small intestine, they can detect these chemicals. And as a result of that, they could release the secretin and CCK. Now we discussed what they do in control of the bile regulation. Next, we wanted to see what do they do in control of the pancreas. So what? So that you don't have to, just making and releasing them even when you don't want to, but you want it to be efficient and you want it to release and sending them when you need them. All right. That's where when the food comes in, the release of these hormones comes in. These hormones is going to go to the bloodstream, reaching the pancreatic cells, and is going to release these enzyme and the bicarbonate. That's what we're going to get into. All right, which is actually very simple. So let's go ahead and get to it. All right. So the CCK induce secretion of enzyme-rich pancreatic juice. What that means is promotes, gives it the green light, says, okay, go ahead and release the enzyme. And secretin does for the bicarbonate, all right, says, okay, green light, bicarbonate, both of which is important, obviously, all right? So vagal stimulation is just vagus nerve. And again, when you're eating and whatnot, the vagus nerve can go different areas and such. As far as the here goes, again, CCK and secretin, and again, it tells you what happens in here, all right? So right here, acidic chyme, what is acidic chyme? This stuff coming from the stomach, as the stomach was acidic, coming in here. But remember, pancreas produce a lot of enzymes. Those enzymes don't work under acidic condition. So we need to neutralize them. We need we need buffer. Our buffer in this case happened to be bicarbonate. All right. So as soon as these cells, enteroendocrine cells that we mentioned, or duodenal gland cells, all right, release the secretin, all right, and CCK, all right. So now they are going into the bloodstream. Where are they going to end up? Yes, they're going to end up in, activating here, contracting here, and, causing the body. We discussed this already. But in addition to that, they have other target cells. Remember, some hormones, they may have more than one target cells. Yes, CCK target cells would be here, gallbladder, and right here, opening up this hepatopancreatic sphincter. But there are other target cells, you see, right? So the CCK, their target cell, also this acini cells, which they can release those enzymes, all right? And the secretin, their target cells also would be the pancreatic cells to produce and release the bicarbonate, HCO3. All right. So uh, end result, here you had a hydrogen ion because you had a HCl, and this was contribute to acidic. So now it's coming. All right. Here you have a HCO3 negative bicarbonate. So again, they can formulate plus HCO3 minus, and now you're going to get H2CO3, which is carbonic acid, which again, at the presence of enzyme, carbonic anhydrase, you could get carbon dioxide plus water. Water is neutral, that's what we want in here. Carbon dioxide, you could expel it, and your respiratory system works and operates that way. You're good to go. All right? So that's the idea with that, right? Without the pancreas, the job of digestion is going to become very, very difficult, right? Now, we are done with the hormonal and control, regulation. We need to go to the large intestine now. So we go back to the process of digestion. We discuss all of these and absorption. As you recall, the last process happens is the defecation or elimination. That's the sixth and the final process of digestion. By this time, you absorb all you could, you digested all you could, but there's stuff that you may not be able to digest, like plant cell wall. A lot of plant cell wall we cannot digest. They're made out of cellulose, and we don't have the enzymes to break down this type of polysaccharides or cellulose, right? Well, they need to be eliminated from our system. Also, excess things that, not everything, a lot is good thing, all right? Because, you don't need any meat for it. Otherwise, you could eat sugar all day long, all right? Why is it that it's not a good idea to have all these sugar all day long? Because you have you have homeostasis to operate, okay? More is not necessarily better. Less is not necessarily better, all right? So you have to be balanced. So a lot of things that you are in excess or perhaps we cannot digest, and they need to be eliminated. And again, that's where the large intestine comes in. There are other job of the large intestine. Another name for the large intestine is called colon, all right? The name came from the diameter, by the way. So, let's go ahead and take a look.
Professor, we can see and can hear you.
I think he got kicked out.
Okay, I see.
Okay, Anthony, you can teach a class.
Okay, wait, what's going on? He got kicked out?
Anthony will teach because he's the host.
Wait, why am I the host?
So you're the teacher now. Continue.
For real?
Why am I recording? That's weird. Hold up.
You better start teaching, man.
Come on, Anthony.
You could do it. You could do better than Professor.
Wait,
what happened to his connection, huh? Talking about everybody get good connection.
I know.
What happened?
For real.
Get good connection, sir.
Legit just thinking that. The irony.
You guys know everything's being recorded, right? Just stop recording.
Anthony, can you stop recording?
All right, so what we left with is the large intestine, and the majority of the large intestine job happened to be water absorption, and let's go to it and take care of the undigested food. So let's go ahead and take a look at bacteria flora. These are the normal bacteria that's supposed to be there. And as I told you earlier, these bacteria sometimes is important to, synthesizing vitamin K, which is important for blood clotting, synthesizing vitamin B complex, things in that nature. But the majority of the large intestine job is to absorb water, all right? Absorb water, and also some of the vitamins, electrolytes, they can be reabsorbed, depends on your needs, water specifically too, all right? So if a person is constipated, that means most likely lacking the fiber in the diet, it's dehydrated, it's lacking water in the diet. That's why you're absorbing so much water from your fecal material. It becomes pretty dry after that, all right? Now, propulsion of the fecal material as they are forming is another thing that happens in the large intestine. And large intestine, as you know, susceptible to, or colon, to cancer. And one of the things that I could talk to you about is, let's say this is the layer of the large intestine. Now, let's say this is the fecal material, all right? When the fecal material, due to lack of water and fibers and a bad diet, are really hard, person in constipated, you know, and so on. Now you require more peristalsis action. So the smooth muscles in the layers that we discuss is involved in peristalsis. They have to work harder, all right? And they can get weakened with aging as the time passes by. So if you're doing the peristalsis to move this forward, you know, the muscles in here, they can get pretty much weakened. So over the time, it might cause diverticula, which is pouch-like appearances, which later on, some of the fecal material can go in there and end up in our blood vessels, and this is known as diverticulitis, the infection. And also this hard material now, the fecal material, as they're going along, you know, they're going to rub against these walls. And there are blood vessels on these walls that sometimes can get ruptured too. So again, you will have internal bleeding. And anytime you're looking at the stool, you can get a lot of information about your digestive system. If you see very dark coloration somewhere along there, it probably indicating that you have an internal bleeding somewhere, all right? If it's dried up, if it's very difficult to come out, if it's taking you a long time, if you see blood in there and so on, that could be a result of hemorrhage. So when the fecal material passing by, they can rub against the blood vessels and it could cause hemorrhage, as you know. All right, and some of these are linked to the cancer. It's not a good idea to have the waste and the fecal material long time in our intestine. That's why fruits and vegetables give you a good bowel movement, allowing it to not to be there for a long time. Some people, you know, they go to the restroom not as often, like maybe once every couple of days. That's not normal. All right, that's not normal. High protein diet, high fat diet, high salt diet, high amount of preservative, high amount of you know, artificial flavor, artificial coloring, food coloring, you know, pesticides in our food. There's so many things that can affect our digestive system, unfortunately, and it is. And now we have to deal with you know, the GM product, the products that are kind of manipulated, their genes, genetic modified food, they're known as. Again, many people think it's safe, it's good to have them, but again, we don't know the long-term effect of it. We may not have certain enzymes to break them. Nowadays, we are coming down with a lot of reactions, allergic reaction towards gluten, towards other things. So it's not a pretty picture, to be honest with you. But if you can have fruits and vegetables, citrus fruits, those are have a high source of antioxidant, and it can help you in terms of you know, processing of the food. All right. So there is a section that I didn't get a chance to talk about. Basically, you have this idea before. I'm probably not going to get into it, but you know, it's just telling you what can be stimulated factors and how what things can be inhibited factor. And in terms of the digestion, you have three phases: cephalic phase, gastric phase, and intestinal phase. What happens at the higher level? Cephalic means brain. Uh, Gastric phase, what happens in your stomach, and what happens in the intestinal phase. All right, so basically is the idea of sympathetic and parasympathetic, right? When you are relaxed, your sight of food, smell of the food, all of those are a stimulatory factor, all right? Your parasympathetic fibers kicked in, and all of those will help you to digest material. Now, at this point in time, you could have more motility of the stomach. Your stomach is probably working towards, as the acidic chyme are being formed, you're processing the food and so on. Again, at the intestinal phase, you know, as the substances or this acidic material is reaching the intestinal phase, you know, that lower pH, and as a result of that, you know, those pancreas starts moving the content, the bicarbonate, because of the hormones that we discussed, CCK and secretin. So all of those are stimulatory factors, but inhibitory factors could be part of it as well. Let's say you have a depression, you have a loss of appetite, you're nervous, things in that nature. You know, when the stomach content is way too acidic, you don't want it to create more acidic on a hydrochloric acid. Everything shuts down. Right? This could be also related to emotional dramas or upset. Your sympathetic activity kicks in, and we learn in the sympathetic activity, basically, you know, all your digestive system shuts down. All right. Same thing with the small intestine and so on. So basically, you have less of those CCK and secretin, and as a result of that, again, those activity won't take place. All right. So that summarizes our digestive system. If you have your phone handy, some of you guys are. Let me stop the recording.